All right. Hello, welcome everyone to today's um oh, I see. Somebody else came. To today's uh, workshop on proteins and protein folding. Now already number 9. Um yeah, but actually for this workshop it's hard to find an end because there's so many amazing things happening in the human body, and it's hard to decide and to make a cut at some point because yeah, there's just so many fascinating things. But I hope, like I mentioned, I think in the announcement, that maybe there will be this one and I guess 2 more of proteins and protein folding, and then we will switch to a different workshop with a different topic, and we'll see how this goes. But I think for today we have a very interesting topic, and by coincidence, it also kind of brings everything together that we've learned so far. And if you've joined most of the workshops so far, it's going to be a very, I hope, like an aha moment where you will kind of understand and be able to use the knowledge that you've gained so far. Okay, but as always, let's start with a little review of what we did last week, right? So, as you remember, perhaps if you were here, last week we talked about nerves and how they work, right? As a central type of cell in our body. We talked about what their anatomy is a little bit, and we talked about how they function. And in the end, as a kind of summary, I showed you this little like interactive program, I would say, that you could play around with. And again, in the beginning, I would like to use this actually to kind of make you remember how these nerve cells work, right? So let me just switch over really quickly. Right, so when you look at this, this is an axon, right? Basically cut. And you can see on the outside is the cell membrane, and you have the inside, and we have a lot of transporters in between them. And these transporters are selective for different ions that are existing inside and outside. If you remember, 2 workshops ago, we talked about basically transport of these ions actively through the cell membrane, right? So that in the end, on the inside, we have more potassium, and on the outside we have more sodium, right? Here represented by the green diamonds or the orange circles, right? And you can see that also you can go closer, see more red circles on the outside and this on the inside. And when we talked about it, we looked at the sodium-potassium pump and how this pump basically uses ATP, which is energy, to transport both ions at the same time in a kind of mechanism that we looked at to basically build a gradient. And if you look at the charge, right, by separating these ions separated by the cell membrane, a kind of electrical potential is created, right? And on the potential chart, I mean, you don't see it now, but it creates a difference in millivolt at around minus 75. So that when a signal comes, what happens is something we looked at last time, right? We had the voltage gated channels, right? Especially the sodium voltage gated channels here in orange. And if the basically the membrane potential increases, right? So more towards the positive, they open up, let all the sodium in, it depolarizes and then repolarizes. And we can simulate that here by simply clicking on Simulate Neuron. And they open up. When the signal comes, sodium goes in, potassium goes out, and then the previous potential is reestablished. Okay, so a very fundamental thing that is going on and is very important for function of almost all organisms. Okay, good. So far for the revision. A little bit talking about this sodium gated channel in detail. Last time we looked at that it's a complex of transmembrane helices, and we had this one helice in there that has a lot of positive charge, and if the outside positive charge decreases, it actually moves up and thereby dragging away these inner yellow helices and opening up the pore. Right? There were some more interesting things. They have some, for instance, also an ion selection area, right? That they can be selective if they, for instance, want to let in sodium or potassium and things like that. And there was also a N-terminal blocker, basically, that shuts off the channel after a short time in order to re-establish the original potential faster than would happen naturally. Okay, oh, too fast. No, okay, I forgot. So, actually, I would like you to switch over to the whiteboard. I hope you didn't remember too much from what you just saw, because today the topic, which brings it kind of together, is going to be vision in the beginning, right? So we are going to talk about how vision works on a biochemical level in this workshop today. So, and in the beginning, I wanted to know, well, what do you guys know? So, because, I mean, the eye and brain and the connection is a very popular topic in normal biology. So, I wanted to know how much detail I need to explain to you, or how much I can skip. And for that, as always, please choose a color and maybe write your name in that color on the whiteboard. And what I would like you to do is, if you're imagining how vision works, how would you put that in a schematic drawing, right? So let's say let's just say we have a smiley, a badly drawn smiley, right? If we then had an eye and the brain, what happens? How would you connect that, right? And I would like you to take a little bit of time, do a little bit of schematic drawing, and then I know basically how much I have to explain to you. All right. So some people joined the board, I think. So don't be shy. I know drawing with your mouse is not that easy. Especially, for instance, if you're using a trackpad. Okay. Pavel. I hope also, Koto, you can help the others out a little bit, because I think you've studied a lot about that in your past project. Anybody else? And we go step by step. We'll see who can maybe add to the illustration. We can also draw everything together. Super chiasmatic nucleus. Okay. Very, very nice. I think nobody knows what that means. Even me, I don't. I mean, I've heard some of those, but I wouldn't be able to describe that. Okay, so, like, first, you have a person, right? And how do we see things? Of course, with an eye. What does an eye look like? Who can draw me an eye? Anatomically correct eye. That's already not that easy. Of course, it can be simplified. There's no complete right or wrong here. Okay. Give a little bit of time because of the delay. Nice. Okay, very good. That's the first part. Right? We have that, which is, right, the lens. Mm-hmm. Don't worry, having multiple drawings is best. Okay. Very good, very good. Very nice. Oh, it's a funny eye you're drawing. But it's correct. It's supposed to be a schematic, right? Very nice. Awesome. Okay. Good, good. Okay, so very nice. Actually, most of you drew almost a little bit better than me, right? Because you're actually also drawing the cornea. So you have, so what everybody has so far is definitely, you have the lens, right? You have the skin on top of it. You have a layer on the back of it, right? Which is called the retina, and you have the nerve that basically bundles the connection to the retina and leads it to the brain, right? Like that. Okay, very good. And in the brain we have the visual cortex. I think it was the back of the brain. I don't remember 100%. I think that's... Pavel, please correct me if that's wrong. Yeah, very good. So does it... Oh, okay. Does that actually explain us how we see? Well, it's clear that basically light comes in to the lens, it's projected on the retina, and the retina basically creates a signal that is then sent to the brain so that the visual cortex can interpret it as a picture, right? And all these things as like depth and 3D and like color and all things like that depend on the brain. But something that we are going to look at today is the retina, right? So because this crucial step of two big black spots. So this crucial step of translating light into electrical signal is happening in this part, right? And of course, since everything in our body works with proteins, this is something that is done by a protein, which we are going to look at today. So let's see, let's go back to the presentation. So, right, schematic drawing of how vision works. And we're going to look at that part. So the retina in detail, as you maybe also have already heard in biology, consists of different layers of cells, right? So usually the ones that are in the front are more for support and kind of nutrition and things like that, and also collecting signal and transmitting it. And then you have the important part, which is basically responsible for the sensing of light and color, which are called the rods and cone cells. Okay. So today we're going to look at also in more in detail what are rod and what are cone cells. Looking at them more anatomically, they look quite strange, but they have some very, very interesting features which actually coincide with something that we talked like many workshops ago, which is that this kind of strange shape is formed by cilia. Right? So we had cilia in a previous workshop, and I mentioned it only briefly. That cilia are also important for vision, right? Among many other senses. In this example, the cilium basically forms this outer segment structure with different floating discs, right? And these discs contain a very, very important part, and these basically the light sensing proteins. So we can see, according to their name, their shape is different. We have the cones that are more cylindrical and get larger towards the base, and we have the rods that basically are just a cylinder and have the same width all the time. So there is a difference, right? So the rods are there basically for sensing light and dark, whereas the cones are there to determining color. Okay, so let's see. So we're going to go more and more into detail, right? So now we know the cells that are responsible for sensing light and dark and vision in general. So what is happening in these cells, right? And the main responsible protein that senses light and dark is called rhodopsin. And we are going to look at how rhodopsin works today, right? And as always, to understand how it works, we are going to use PyMol, right? And on the left side, we have the two numbers that we need. Actually, I forgot. I need to write them down as well. F8 UR. Very good. So let me switch to PyMol. Okay. Very well. Okay. Also, if you're using PyMol, let me know. Again, that helps me determine how fast and how detailed I should explain what I'm doing. I hope at least one of you is. Because it always helps to understand it easier if you're able to freely determine, for instance, the angles and what parts to look at. And it also builds a certain skill that you might perhaps need later if you decide to go into biochemistry or not. Okay, Pavel is using. Well, Pavel, you're basically an expert, so I don't have to slow down too much. Come on, guys. Okay. So as mentioned, the two proteins, right? Yeah. The two proteins that we need today are 1F88. So we're going to type in fetch 1F88. Very nice. And we also need another one. It's called fetch 3PQR. All right. So all three proteins that I just loaded into PyMol are basically the same, right? The same protein, just different conformations. Actually, two different conformations. Something to mention is that recently, like a little bit a newer finding, was that these proteins usually they also exist in dimers, and that also helps them a little bit with their function. But we don't need to go into detail about that. Maybe something that is worthy mentioning that this is also something that has been known for more than 20-30 years, right? How this whole vision functions and what kind of molecules and proteins are involved in that. But step by step, with newer techniques, for instance, like X-ray crystallography and cryo-EM microscopy, we get a more better and detailed and maybe also more illustrative way at looking at these kind of proteins. So let's see. So again, I told you this is a dimer. We want to do something which is basically the same as we always do. We want to align both proteins, or both not isoforms, both conformations. Right. So, and what you see again is that, well, dimer, we don't want to align two onto one. So what we have to do is erase one of it. So we go to display the sequence, and what we're going to do is we're just going to select the second half, and we're going to delete it. Just right click and remove. So there are a bunch of things that are left over, right? Some molecules that we can simply, actually let me select from up here. All of these we can just right click and remove. Also on this one we don't need those labeled NAG. These are also, simply we can remove them. Same goes for the other one. In this case, we called also NAG, BMA, MAN, and all sorts of things. We don't need those. Very good. Actually, Give me a second, I'm going to close the window because maybe it's loud. Okay. So let's see. Now we have both confirmations and only one of them, right? So similar to what we always did, we click on A for action, and we going to click on align to molecule, and 1F88. Right, and that went very quick. It's actually quite a small protein. That's why your computer does not have to shift select by mouse. Shift select. Instead of dragging, yeah, that's true. Right. We center our view and we can see the protein, right? So this already should look very familiar to what we've seen in the past, right? Because a lot of the proteins that we've been looking at, for instance, like transporters and things like that, they were all membrane proteins, right? So membrane proteins means that they are located in the plasma membrane, and they usually have common structural features that you can find. In this case, maybe if you remember from last time, the sodium-potassium channel had 6 alpha helices. This one has 1, 2, 3, 4, 5, 6, 7. 7 alpha transmembrane alpha helices, right? Very, very common, especially for pores or transporters. Okay, very good. All right, so now we want to look at it a little bit closer. So we see there is an N-terminal part that's sticking out on the outside. We have something on the bottom that's extra and loosely arranged. And we see that there's also a lot of conformational change going on in these alpha helices. So we've been looking a lot at proteins, and most of the function and conformational change comes from the properties of amino acids, right? So the change in how these amino acids associate with each other leads to then, for instance, like drifting apart, coming closer together, and so on. But something that proteins usually cannot do is sense light. Okay, so also in this protein, right, there's something that basically the protein uses to sense light, right? So if you think about it, what is light? Light is an electromagnetic wave, right? Usually also called a photon, right? If you're thinking about a single package of energy. So in order to sense this, right, you need to basically create a change in conformation in the protein, right? But the only way you can do that is if you have a chemical that is sensitive to light. In this case, this chemical, what I'm talking about, is right in the middle here, right? And to make it more visible, we are going to give it a different color. Maybe one we can make red, and the other one we can make magenta, right? And yes, it sits very central on this protein. Actually, for a little bit for explanation, let me switch back to the presentation, right? Okay, so this is the chemical that I'm talking about. This molecule is able to change structure by absorbing the energy of a photon. Right? You don't have to know about so much about chemistry to understand what's going on. A little bit the name tells you about the conformational change, but all you need to know is that something goes from bent to straight. Right? So this is a long chain molecule, and in its usual form, it is bent, right? And upon absorption, it straightens out. In more chemical terms, you would say it goes from a cis conformation to an all trans conformation, right? So, which means cis means how this arrangement of bonds is in this molecule. So you have like at position 11, you have a bend in it. This is what the name means, 11 cis. And trans means if it's like a zigzag like this on the bottom. So cis and all trans, retinol. And, well, maybe you've never heard of retinol, but I bet you've heard of something that I'm going to mention right now, which is called vitamin A. Right? So everybody's heard of vitamin A, and you've probably also heard that vitamin A is supposed to be good for your eyes. So, but what does it actually mean? Why is vitamin A good for your eyes? Does it make you see sharper? Does it give you night vision? Does it, I don't know, what does it actually do? And here I want to give you the answer to that, I wouldn't say urban myth, but maybe misunderstanding. Because something that you know, for instance, that exists in many vegetables and also fruits is a color molecule, which is called beta carotene. Right? Beta carotene is usually red-orange in color, and also a little bit yellowish. And beta carotene is basically a long chain that looks very similar to our retinol, right? So, simply put, this beta carotene is two retinols linked in the middle together. So, when we eat vegetables like carrots or tomatoes or other things that are red, we take up a lot of carotene, and our body uses this. It basically splits it in the middle. It adds an O at the end, and we have retinol, which then goes into the eyes and is changed into our 11-cis retinol. So, does it mean that eating a lot of carrots makes your vision better? Well, if you have vitamin A deficiency, yes. If you're a healthy human being, no. So, in your eyes, you definitely need cis retinol, and there has to be a common supply or a constant supply, I mean, of vitamin A to keep up levels of this retinol in the rod and cone cells. However, eating more carrots than usual will not increase the level significantly. And if there's a saturated limit reached, having more of these does not improve your vision. So it does not basically, for instance, like maybe some of you believe if you eat more carrots, for instance, you won't need your glasses anymore. Or perhaps you will see better in the dark, right? So as long as you're eating healthy, you're eating vegetables once in a while, and you're eating, for instance, also meat. Meat also has a lot of vitamin A. You will have a sufficient supply of beta carotene, which is transformed into retinol, which is then used to sense light. And how this happens, let's go back to PyMol. Okay, so looking at PyMol, this is what we just talked about, right? So right in the middle. So we can see actually two different versions. One is the straight form in red, and the other one is the bend form in magenta. Right? So this is the retinal, which is used. So usually the initial state of rhodopsin is with retinal in the bend form. Then when it when it receives a photon and the energy to straighten out, it is connected to many other amino acids around it and held in place. And if it changes conformation, also these amino acids change. And this change, like in many other examples that we saw, is propagated throughout the whole protein. And let's look to where it is propagated to, right? So looking around the protein, where is the biggest difference in structure that we can see between the green and the blue? Right, so take a little bit of time, look at it, and you can see there's a little bit of change in the top, right? But the main change that is happening down here, right? So let me focus on it. Is happening here. And so these alpha helices are basically moving outward, moving a little bit apart from the inner membrane section of this protein. Right? So from this to this is the whole transformation that's happening upon light hitting this protein. So, but something that I have to mention, this is not a channel like we looked at in the previous workshops, right? So this is not an ion channel. So if that changes, if it moves further apart, there's nothing coming in as which would create a signal. To understand why this change has an impact and makes us sense light in general, we have to take a better look at what is happening in the bottom part of this protein, right? So going back to the presentation. Let's see. So fortunately the pictures are not very high quality, but it's basically the summary of what you just talked about, right? So the change from this bend to the straight form leads to a change in this alpha helix, basically moving out, moving downwards. And what that does is, if it leads to a change on the right, right? So if you look on the right, the top part is basically the protein from the left, the rhodopsin. On the bottom part, we have what is called a G protein. Now, G protein are a little bit complicated. They're found in many parts of the cell, and they have many different functions. In this case, this one is called transducin. I think transducin. So it has 3 different parts, right? So orange and light blue. And upon this alpha helix moving apart, this has actually a place to bind. So as soon as this changes, this can bind to it and form this type of complex. So the following change is a little bit complicated, but I hope I can convey it to you so that it makes sense. So, of course, I told you before, light is translated into an electrical signal, but it is not as direct as you might imagine. So, what we just looked at is the first part here, right? So light comes, and the inactive rhodopsin becomes its active form, right? And the G protein, transducin, is able to bind the bottom part. It then splits off one of its parts, because it has 3 different parts. One of it is able to split off and binds to a protein called a phosphodiesterase. This then again relays the signal to a signal called guanidine monophosphate, and it's a cyclic version, cyclic guanidine monophosphate, and this is something that should remind you a little bit of what we talked about in the last workshop, I believe. So we talked about passive transport, and we talked about ways passive transport, like ion channels, can be triggered. Right? So we had different versions. Last time we looked at voltage. But we also had transport, or like these kind of channels, that can be triggered by signals, right? And this signal, one of these signals can be cGMP, right? cGMP is produced by this phosphodiesterase, and upon binding to a sodium channel, sodium is able to go in. Upon binding, actually the sodium channel is closed, right? So, and by closing, it leads to hyperpolarization and thereby to a signal in the end. And so a little bit, a very long indirect chain from actually the activation of the protein to then a change in the voltage along the membrane, and thereby emission of a signal, right? But I hope it kind of makes sense, and it a little bit brings together what we've talked about so far, right? There's also something I have to mention, because it explains a very interesting feature of our eyes. Well, not feature, I would say like a phenomenon in our eyes. So, well, this might look a little bit confusing, but unfortunately that's the best schematic of what is happening that I could find. But let me explain to you step by step. So rhodopsin, right, is basically the name for the loaded version of the protein. So the one that has the retinol in it, right? If it does not have retinol in it, it's just called opsin. So after light hits rhodopsin and the conformational change, what happens actually is the retinol leaves the protein, and it becomes opsin. And it has then to be loaded again with the molecule that is bent again, right? So because it has to return to its initial form, but in the rhodopsin itself, the retinol does not just go back to its bent shape. That has to happen on the outside, right? And that happens in what's called the I'm not 100% sure what RPE means. I have to guess right now. I would say R stands for retinol, P stands for pigment, and E stands for epidermis, maybe? Basically, a layer of cells below the rod cells, right? That are specialized into recovery of this band shape of the retinol. So basically it goes out, it goes into a different cell, it has to be recovered, goes back into the rods, and there then goes into the opsin again, making it active again. So quite a complicated system that nature has thought of there. And it also leads to some very interesting phenomena. For instance, have you ever noticed that if you look at a bright light for long, you go blind for a little while? All right, so why does that happen? And why this happens can be explained by this, right? Because it's complicated, it actually takes quite a while to recover rhodopsin. So what happens if you just are exposed to a little bit of like usual, means that some of the rhodopsin is turned into opsin. Just a part of it, right? So some active form remains and can be hit by new light after a little while, right? And thereby you can still see light and dark. But if you receive a lot of light all at once, what happens is that all the rhodopsin turns into opsin, right? So what does it mean? So that means that all of the opsin needs to be recovered and turned into its active form. But as I just explained to you, that's quite complicated, and that actually takes time. Whereas this whole process takes mere milliseconds. The whole recovery can take minutes, right? So basically this is called rhodopsin bleaching, and if you bleach all of the rhodopsin at once, you won't be able to see anything. Well, at least not light and dark for a couple of minutes, right? It depends on how strong the flash is and how much light you are exposed to. Of course, also if the light is too strong, it might actually damage the cells, right? Because still light is electromagnetic wave and carries energy, and if it's too strong, it can have a lasting effect. So please don't go and try this out, is my recommendation. But I guess everybody of you has made this kind of experience before. And I kind of think it's neat that you can now explain it, right? Instead of just wondering why. Also something that I want to mention, because what I just told you about the rhodopsin mainly involves seeing light and dark. And, well, what does light and dark mean? So, actually the rhodopsin is sensitive to a certain wavelength. This wavelength is not does not span the whole spectrum of visible light, right? So it's mainly located at 498, which responds to yellow color. But at the same time, it's basically where most of like the blue, the green, and the red mix together. If you want to analyze the cones, basically, which we saw in the beginning, cones are there for looking at color, right? And color can be separated by different wavelengths. And in this case, it's not rhodopsin, but something that is very, very similar, right? So in this case, they have different names. It's called S-cone opsin, M-cone opsin, L-cone opsin in humans, right? So some of them have mainly this one, which senses the blue, which senses the green and the red. But the mechanism is the same for all of them, right? What is different in them is the protein, right? So the opsin, that's what has a different name. Something that is changed to basically perceive different wavelengths of light are different amino acids in the opsin, right? So you basically just have like minimally different versions which are able to absorb the light energy more efficiently from different wavelengths. Right? And this light absorption then leads to the straightening and so on, creating the signal. Right? I hope that makes sense. And also something, maybe also an interesting side fact. So if you look at this, and if you look at that, I don't know if you have seen this before. Those are so-called night vision goggles, right? So if you've seen maybe some war movies or special agent movies, whatever, like James Bond, you see them once in a while, right? So basically goggles that make you able to perceive or see at night, usually used by the military, and the signal that they receive is translated into something like this, right, on the right, which is a brightly green picture. So why is it lightly green, right? Why is it not red? Why is it not blue? Or any other color? Right? Well, the answer is because the eye is most sensitive to a yellow-green, right? Because it uses basically an overlap from all different opsins, right? So in addition to amplifying the strength of the light by the camera, also the picture that is then generated is not supposed to blind you, and the very low light that is used in yellow-green because your eyes are most sensitive to it. Also a neat little fact. Okay, so before I go to the next part, which I wanted to talk about, which also is a part where it all comes together, are there any questions so far about how the biochemical part of vision works? Because I kind of want to make sure that you understand how all of this works before moving on. Because the next part is a little bit building on top of that. Any questions? Okay, so I hope the delay has not become too strong or too long till now. Oh, I see, Pavel. You're always with your quantifying questions. That is very... I would have to Google that. Yeah, maybe that's interesting, but I usually I'm not looking for actual numbers when I talk about things like that. I think it can be varying though among people. There's definitely a big discrepancy between different animals, of course, because we have, for instance, animals that can see at night, right? They have a lot of more rhodopsin and more mechanically also different eyes to amplify the light sensitivity. For instance, if we think about cats, right? Cats have like a reflecting layer actually inside of their eyes, which bounces the light back and forth to amplify it inside the eye. That's why if you shine a light into a cat's eye at night, it will actually reflect the light towards you and look like a demon. So if you ever had a cat or seen a cat at night, you know what I'm talking about. Yes, but also the amount of rhodopsin definitely plays a role, but I don't know how much. Okay, very good. So if that is clear so far, let's move to the next part. And I hope also, Pablo, you can assist me perhaps a little bit, because you're the one that suggested it. But I think it's I have to admit I haven't heard of it before. Actually, you mentioned it to me. But it's nevertheless really, really interesting. It's called optogenetics. Right? So the title is, or the name, I think, is a little bit misleading. It should definitely tell you something from the first part, right? Optics, right? Has to do something with vision. And well, the second part is, well, it has to do with genetics, genes, right? Something like that. I guess that makes it a little bit clearer if you hear it. So optogenetics started by basically looking at different kind of microorganisms and how they sense light. So we have on Earth, we have many different organisms who can sense light that do not necessarily have to have eyes, right? Or yes. So I mean, most of them that we know usually do have eyes, specialized organs to sense light. But how they work can also be very different. So for microorganisms, they also have very interesting mechanisms of how they sense light. Maybe you've seen some of them before, right? So the left one is a green algae called Chlamydomonas. I'm actually not sure what the bacterium in the middle is, and the right one is Volvox. Is also a type of multicellular organism which does photosynthesis. So all these organisms, they have proteins that can sense light. Usually, I mean, what we looked at right now is a way to sense light, right? We had rhodopsin in humans. For microorganisms, they have other ways to sense light, and they can be categorized into three different areas. Ones are photosensitive, right? So, for instance, they can be channels that open when light is present. It also can be something that's called light-induced dimerization. So basically, exposure to light changes the structure of a protein X, which then changes its conformation so much that it can bind to a certain binding partner and therefore create a signal. Or there's something called also photocaging, where basically an active site is hidden within the protein, and then upon light exposure, it opens up, exposes the active site, and then produces a signal. Right? So what we're going to focus here is mainly going to be the left one, right? Inducing channels with light. So it's a very interesting feature. And this feature was first found in the green algae, Chlamydomonas, because this microorganism actually has something that's called an eye spot. So we know that it actually can sense light. So if you would grow a bunch of them on a plate, and you would only expose one side of the plate to light, well, over a short term, all of these algae would slowly move to only the one side of the petri dish. So it all has to do with the flagellum, and how it is regulated by the eye spot. Right? So the eye spot, as it says, is like an eye, and it senses light and dark. Upon looking at how this works, they found, well, it is an eye spot because it has a different color, and this different color is basically there to reflect and to intensify the light that is basically shining on the algae. And it is then sensed in the membrane by a protein that is called channel rhodopsin, right? So it's called rhodopsin, but it regulates the channel. So it's very, very related. But instead of the complex and difficult mechanism that we just looked at in the eye, it's kind of simplified. Right? So because, well, it's only one cell. There's no other cell to, for instance, recover the retinal, things like that. So it has to evolve a different strategy to deal with sensing light. And it does so by directly controlling a channel for ions with light in this eye spot. And then upon sensing light, it basically can change the rotation of its flagella and thereby move into one or the other direction, towards or away from a stimulus. So I don't want to go into Python again with this. I just want to show you that it's actually very, very similar to the rhodopsin that we just looked at. Right? So we have 7 transmembrane helices, right? So 1, 2, 3, 4, 5, 6, 7. And we have a rhodopsin, which is bound in the center of it. We have an end domain and so domains that are outside and inside of the membrane. And we have a significant change in 2 alpha helices that are basically moving away from the whole protein, right? So what this does, if you look on the right, this whole protein, those 7 alpha helices, they form a tunnel through the plasma membrane. Right? This tunnel was calculated by a group and is shown as this kind of like deformed bubble. So if you look at that, you can basically then understand how, if these, for instance, this TM1, right, which is moving in TM2, right, TM1, TM2, by them moving apart, they're basically opening this blockage for ions that would exactly fit in here. And ions, again, are attracted to this pocket because, well, it's very electronegative. There is a little bit of difference, right? So, as I told you, it also uses retinol, but it actually has a way to recover the bend form. So it starts off in a straight form with energy bends and then recovers itself again. Which I'm not sure, I haven't been able to find why, but I'm sure probably somebody has explained by now how this works. So how's that applicable, right? So cool, you find something that reacts to light and opens a channel, right? And from all the things that I've told you so far, that should have maybe given you this type of aha moment. Aha, where do we know channels that let through ions to produce a signal, right? So if you're thinking, well, nerve cells, then you are correct. Right? This is exactly what the idea that researchers had. Well, we have a channel which we can open with light, And we can basically, with light, then initiate basically a electric signal in a nerve cell, right? And that's amazing, right? So usually what's happening in nerve cells is you have an electrical signal and all of them that are connected turn on. Well, if you would just shine light on them, well, that wouldn't do much, right? But if you had, in addition to what we talked about, the sodium, the voltage-gated sodium channels expressed in these cells, you could shine a light on them and they would turn on without an electrical signal. And that was a very, very a great idea. And I think it was like announced basically the method of the decade, or the first decade of the 21st century, because it has so much potential, right? It has so many potential applications, and once a lot of the technical sides are overcome, you will see by what I will show you in the next slides. This will be the future, perhaps. Okay, so first examples of how this is applied or has been applied to different animals or in different scenarios is, well, you have to get this protein, right, which reacts to light, you have to get that into nerve cells first. Which, maybe if you've heard me talk in the past about maybe how we use like DNA and put it into cells to then have it express in these cells, there's a way to use basically viruses, lentiviruses, to stably express certain genes in cells. And you can use these to basically make nerve cells that express this channel rhodopsin. And here is what happens, right? So on the right, I will show you a short video of a model organism called C. elegans, which is a worm with a defined number of cells. And basically what researchers did, they just gave it this channel, right? And then what they did is they used green light, shone it on this worm, and looked what happened. Oops, that is not supposed to happen. Okay. So let's see. So what's written down here is basically the speed of the worm, right? So in the green is when the light turns on. So it's moving happily, and then light turns on and stop, right? Starts moving again and stop, right? So what they found is, so it actually works. We can put this channel into an organism, we can shine blue light on it, or green light, and it will react, right? So something in the signaling within its nerve cells changes, so it is not able to move anymore, which was the very first success, right? So after that, well, how would we do that maybe in a more complex system, right? So the idea is, for instance, in a brain, right? So we have this channel expressed in the brain. We choose a single neuron. We're going to shine some light on it, right? So with this the upper one is the light, and what we want to see is a signal as a response, similar to what they saw in the worm. So there's a way to do that, right? And over time, they've been able to do that in mice. So first you have to make this genetic construct, then you put it into a virus, you inject this virus into the brain of the mouse, and then you put a fiber optic line into the brain of the mouse, and then you can shine a light in different areas of the brain, which is amazing because this is not really something that you can do with any other technique, right? So any other techniques that you can think of are very, very invasive, or at least a lot more invasive than this one. And let's see. So this is what it basically looks like in a real mouse. And how this works, instead of me explaining to you, let the person who actually had a huge part in developing all these techniques and all the knowledge around it, have him explain it to you how this whole thing works, right? So let's see. We'll take about 5 minutes in the video. It's very, very interesting. Oops, no, not from the beginning.
Simple, but it took a number of years to actually say we had finally discovered how to make this work. Part of the problem was, how do you get light into big mammalian brains? And this is an early sketch of Feng Zhang, where he designed the fiber optic interface, and this was from 2006, but then this looks very much like how we now do it today. So you can see he's a good artist as well as a great scientist. And this fiber optic interface has since allowed us to do many things, but I'll show you one other first experiment. This was 2 years later in 2007. Feng was working with some mice that we had made in collaboration with Guoping Feng and George Augustine. And these were mice designed to have channel rhodopsin expressed in the brain. And what Feng did was he implanted into this mouse a fiber optic on the right side of its brain, which, as you know, will control movement toward or attention to the left side of the world. And when he turned on the light, as you'll see here, the animal immediately starts moving and starts turning to the left. And this was the moment when I would say we finally knew that we had made something that was going to be broadly applicable, generalizable. And that was followed quickly by a collaborative set of experiments with Luis de la Sella, Alex Arabanis, Antoine Adamantidis, and others, where we applied this fiber optic interface to target a deep structure in the brain in the lateral hypothalamus. Feng designed some promoters that would fit into a lentivirus and allow us to control just one kind of cell deep in the hypothalamus. And in this work, we were able to play in certain kinds of activity and show some of the neural codes that were causal in sleep-wake transitions. And then, even then, though, this required, a very specific fragment of a promoter, the hypocretin promoter, which would fit into a virus. And there was still, rightly so, some skepticism as to how broadly generalizable this would be. But between 2009 and 2010, we finally were able to show that there were a broad array of different tools you could use to generalizably target different kinds of cells. And by this point, I would say by 2010 or so, people were able to see that this could be applied to virtually any system. A good example of how that all worked was in our studies of anxiety. Now, anxiety is a normal part of life. It's a healthy part of life. Of course, it can become excessive and it can be pathological in some cases. But what we were able to do is to use optogenetics to play in patterns of activity or suppress patterns of activity in particular cells and projections in and around structures relating to the basolateral amygdala and the bed nucleus of the stria terminalis. And the way we did some of these experiments is we injected a virus, an adeno-associated viral vector, into the structure of interest, the BNST, but then we were able to position our fiber optics in other downstream structures. And by doing so, we were able to recruit cells defined by having a particular projection pattern, which, as you can see, is a generalizable strategy. You don't need a special kind of animal or a special promoter. You just need to know your anatomy. And this works because the opsins are trafficked down the axons of the cells, particularly if we help them out with little trafficking motifs that were discovered in the lab by Viviana Gradinaro and her colleagues. And I'll show you an example of how this sort of thing works. This is an experiment. I'll play this movie. Kate Tai in the lab did this work, which was published in 2011. This is what's called an elevated plus maze. The so-called closed arm has these elevated walls, and you'll see that the animal prefers to spend its time in the closed arm of the maze, very rarely venturing out into the open arm, which is like walking the plank for an animal. But in our studies of anxiety-related behaviors, what we found and what Kay was able to show, and we followed this up with a great deal of other work, was that there were anti-anxiety pathways buried within the brain. When the blue letters come on here, you'll see when she started driving one of these, as we discovered, anti-anxiety pathways, and you'll see for the first time the mouse is willing to go out into the open arms and explore them, which it never had been before. Nothing else changed about the animal, the speed or anything else. It simply was willing to accept this risk, and this was instantaneously reversible. And this, of course, is only one feature of anxiety, apprehension in the absence of immediate threat, but there are other features too: respiratory rate changes, subjective feeling. Anxiety feels bad, and resolution of anxiety feels good. And in later work, we were able to show that different projections coming from the bed nucleus of the stria terminalis recruit different features of anxiety using this projection targeting approach, where we position the fiber optic in a different location than the injection of the virus. And this sort of thing allowed the generalizability of optogenetics to become clear. And so in this sort of work, if you think back to those very early humble experiments in culture, you can see it took a number of years to finally put all the pieces together. I'm often asked,
All right. So, as you can see, pretty amazing stuff, right? So he only showed it for one example, right? Which is, for instance, movement, right? Induced by just shining on one hemisphere. Or he showed it even in the context of anxiety, right? In mice. So, needless to say, this kind of research can be done for all different kinds of behavior, right? It can be done for memory, for all parts of the brain, to analyze with causation if this is active, what changes, right? Which is a technique which, yeah, something that has never been able to be done before. Okay, very good. All right, so yes, so which mainly focuses on the left part. Of course, there are also other ways these optogenetics are used. Right, it doesn't always have to be the brain. There are all kinds of different signals that could be triggered by light instead of others, right? So, but we don't have to go into detail about those. And yes, I hope with that I could show you an interesting and modern application of all the knowledge that I've showed you so far. And if you have some questions, please feel free to ask me or Pavel, I guess, because he suggested it to me. And otherwise, I think this is all that I want to tell you for today. So I'm going to give you a little bit of time. I'm going to wait here and read the chat and answer some questions if they come up. For those of you that go off now, I hope I can see you next time. The next 2 times, I hope. All right. And for the others, I'm here to wait. Okay, very, very good question, Pavel. Well, to stimulate the human brain, it's you would have to create an interface between basically that allows us to shine light into certain parts of the brain, or you can also, something that has been done in the past very, very effectively, you can also engineer the channel rhodopsin, right? For instance, to be sensitive to different types of wavelength, right? So it doesn't have to be perhaps light. It can be a lower or higher wavelength to stimulate it. And maybe, I'm not 100% sure. I think it would have potential to be triggered by other types of electromagnetic waves, not only visible light. Also, of course, if you know more, feel free to add to what I said. I see. Yeah. So yeah, something that I didn't show you in the video is that, well, the person here, he's talking about, well, how can this be applied to the human brain? And he's talking about magnetic wave stimulation, which has been shown for, I think, cocaine addicts when he talks about it. That if stimulated in a certain part, which has been tested in animals before, the craving for the drug cocaine is significantly reduced, which due to magnetic stimulation. I didn't know that this was actually considered to be optogenetics, because, well, the genetics part is missing and also the opto part. So you're not using light and you're not using anything that is genetically engineered in the brain. All right. Magnetic genetics. Okay. All right. So if there are no further questions, if you would like to know more about this type of field in science, please let me know. And if you have any questions, always ask us. And also, if you have some things that would be interesting for you to, for instance, know about how does this work on a biochemical level? How does this actually work in detail? Please let me know. I'm always very happy if there's any input about things that would interest you to learn about. Okay. All right. Then I guess I will see you guys next time. Bye bye.